Well, good afternoon and welcome to today's webinar looking at science and technology demonstrations launching on board SpaceX's 31st Commercial Resupply Services mission to the International Space Station contracted by NASA. I'm Patrick O'Neill, Public Affairs and Outreach Lead for the International Space Station National Laboratory, and I'll be your moderator for today's event. Okay, so let's talk about logistics for this launch. First and foremost, as mentioned, this will be NASA's SpaceX CRS-31 mission to the orbiting laboratory. Launch is slated to take place from Launch Complex 39A at NASA's Kennedy Space Center in the coming weeks. So please be sure to follow various NASA social and online platforms as details emerge shortly. SpaceX's Dragon spacecraft will be filled with more than 6,000 pounds of critical research and supplies and will launch on a Falcon 9 rocket as part of its approximately 30-day mission to the space station. As you might have heard, I did say the magic word, research, which is why we are all here today. Today, we are focused on just a few of the investigations launching that will be supported by the on-orbit crew during this mission. We have an exciting lineup of speakers showcasing the diversity of experimentation that the space station enables. These representatives will provide an overview of their project and answer questions from you, the media, who have registered in advance. And we thank you for doing that, by the way. Okay, before we hear from the teams launching investigations on this mission, we will first hear from the ISS National Laboratory as well as NASA. For media, after these remarks, I will open to any questions. To ask a question, please use the raise your hand feature within the Zoom platform, and we will unmute your line so that you may ask your question. Or you can also ask your question within the Q&A box. All right, I think that should put us in a pretty good place from a logistical perspective. So what do you say we start talking about space-based R&D? To kick off the conversations, I would like to invite Dr. Kristin Kopperud, Science Program Director, Research and Innovation for the ISS National Lab, to provide some opening remarks.
Thank you, Patrick. I'm very excited to be here with you all today to highlight some of the great research and technology demonstrations launching on NASA's SpaceX CRS-31 mission. As Patrick mentioned, I'm with the ISS National Lab and had the privilege of working alongside some fantastic researchers and hardware partners to leverage the space station to its fullest potential, bringing value to humanity through space-based research and enabling a sustainable market economy in low Earth orbit. It has been an incredibly busy year for the ISS National Lab, and considering this will be the last resupply mission to the space station in 2024, what a great display of investigations to finish off the year. This year alone has seen more than 100 payloads sponsored by the ISS National Lab launched to station, representing hundreds of experiments across a wide array of disciplines. NASA's SpaceX CRS-31 is a great representation of the diversity of experimentation taking flight that our on-orbit crew will support. We have life and physical science investigations on this mission, technology development, materials science, and student-led projects as well. While I realize we have multiple investigators who will highlight research sponsored by the ISS National Lab this afternoon, I thought I would mention some other projects also slated for flight. First, I'll highlight the Bristol-Myers Squibb will build on its legacy of protein crystallization on the space station with a project seeking to crystallize model small molecules. Crystals grown in microgravity are often larger and more well-ordered than those grown on the ground and could have improved morphology. Results from this investigation, in collaboration with ISS National Lab commercial service provider Redwire Space, could support the manufacturing of more effective therapeutics. For nearly a decade, the ISS National Lab and the U.S. National Science Foundation have provided yearly funding opportunities to further fundamental research on station in both the life and physical sciences. This mission has 4 NSF-funded projects flying, including one from Oregon State and Texas Tech focused on cardiac health. This experiment will use 3D printed cardiac organoids to study microgravity-induced heart muscle atrophy. Results could lead to increased understanding of the condition, which occurs in several illnesses, including cancer, muscle disease, muscular dystrophy, diabetes, sepsis, and heart failure. There are also a variety of student-led projects on this mission, including more than 35 associated with the Student Spaceflight's experiment program. This program aims to inspire the next generation of scientists and engineers by actively involving school communities in the development of scientific investigations leveraging mixed sticks on the space station. A variety of the projects from the students on this mission will be focused on plant biology, specifically a variety of potential food supplies for astronauts on long-duration spaceflight missions. Next week, the ISS National Lab team plans to put forth a variety of press releases and content around the upcoming launch. So be sure to check back on our webpage at issnationallab.org or follow us on X, Facebook, and LinkedIn to hear more about these projects as content goes live. In the meantime, I'm very excited to hear from some of the research teams flying projects on this mission. Thanks, and back to you, Patrick.
Awesome. Well, thank you very much, Dr. Kopperud. To your point, a lot of exciting stuff that's flying on this mission. And again, as you also alluded to, in the next couple of days and weeks, we will also be having a variety of features, videos, and other fun content that will be talking about some of the projects sponsored by the ISS National Lab on this mission. Next up, I would like to welcome Dr. Meghan Everett. She is the Deputy Chief Scientist for NASA's International Space Station program to provide some perspectives from our nation's space agency.
All right. Thank you, Patrick. And as always, it's a pleasure to be here and talk with you guys about some of the science that NASA is doing on the International Space Station, and that is being brought up specifically on CRS-31. So Kristin did a tremendous job highlighting all of the great science that National Labs is doing. I'm going to talk about some of the science on the NASA side, but before I get into that, for this audience, I also want to highlight what an amazing engineering and scientific feat the National Laboratory on ISS is. So it is the only place where we can look at the effects of the extreme space environment, such as solar radiation, microgravity, and a variety of thermal ranges on materials, human cells, the human body itself, and even for some rodent research. So again, this is just a tremendous capability, and all of the science that we do on the space station brings innovative and unique responses that enable us to explore beyond low Earth orbit in the future. It brings back results that are impactful for humans here on Earth, and can even help us better understand things like climate change and space weather. So in this flight, we have investigations from NASA, our international partners, and national labs, which you've already heard about. So not only is the space station an amazing engineering and scientific feat, it is also an outstanding example of how we can collaborate across nations, industry, and academia. So some of the investigations you're going to hear about in the webinar today from scientists. I'm going to give a quick highlight here, and then I'm going to hand it off. They're going to get the opportunity to talk about their science in far more detail than I can do. But some of the ones we want to highlight here, first is the Coronal Diagnostic Experiment, or CODEX. And this is research from NASA's Goddard Space Flight Center, Korea's Astronomy and Space Science Institute, and the Italian National Institute for Astrophysics. So again, this is a great example of how we collaborate across different organizations to do science on the space station. And you'll hear from Dr. Newmark later, but just for a little enticement to get you to stay on for the rest of this webinar, Corona is a solar coronagraph that blocks out the really, really bright light from the sun to allow us to get more details and a better look at the sun's atmosphere, or its corona. And so this study is specifically looking at the heat and the solar winds, or the solar winds that come off of the sun, and how fast they travel and how hot they get. So Dr. Newmark is going to be able to explain that much better and in greater detail. But this is one of a really fascinating study that we're really excited to see the results on this going forward and how these come out during this flight. Another study I want to highlight is the ANT1 radiation tolerance experiment with MOS in orbit on the space station. And rather than say that big long word, we call it Artemis. And Dr. Zupanska is going to talk about this in more detail later in this webinar. But again, just to give you a little highlight, this investigation uses moss found in Antarctica that's tolerant of extreme conditions, and it has roles in oxygenation, regolith enrichment, and even potentially medicine production. So this is very pointed at furthering human exploration beyond low orbit, where this moss could potentially help with bioregenerative life support systems, and even help support plant growth. So while this is really focused on, from the space-based benefit, looking at exploring beyond low Earth orbit, we're hoping that some of these results in this science can be brought back to Earth to improve crop growth and even environmental cleaning systems here on Earth. So again, tremendous impacts from this study, and we can't wait to see the results of that. And the last one I'm going to highlight is called Euro Materials Aging. And this is brought to us by our European Space Agency partners. And this study looks at how space affects certain materials, including those that are used in optics, insulation, and 3D printing in space. And the results of how these different materials can withstand the harsh space environment will help us better understand and how to design spacecraft and satellites, and how they can they survive in this very extreme environment of space. So with that, I'm going to hand it back to Patrick, and really looking forward to hearing from the rest of the speakers in this podcast.
Well, thank you very much. So at this point, if there are any questions from our friends in the media, please feel free to either raise your hand or ask a question in the Q&A box. But if there are no questions, then we will jump right on into some of the researchers for today's webinar. Perfect. Okay. Well, with that in mind, Dr. Everett did allude to this investigation, but we will hear straight from Dr. Jeffrey Newmark, who is a research astrophysicist for NASA's Goddard Space Flight Center, to discuss the Coronal Diagnostic Experiment, or CODEX, which again is using a solar coronagraph to examine solar wind and how it forms. Dr. Newmark, how are you today?
Oh, good. Thanks very much, Patrick. And thanks, everyone. If you can bring up maybe the first image. So let me talk a little bit about CODEX, the Coronal Diagnostic Experiment. We've been studying the outer atmosphere of the sun. We call that the corona, and the origins of the solar wind, the stream of particles that leave the sun and fill all of interplanetary space. We've been studying this for decades, and still there are a lot of mysteries. We just don't yet fully understand it. We have some really basic questions. What heats the corona? The surface of the sun is of order 10,000 degrees. The corona is 1,000,000 degrees. Where does all this energy come from? The solar wind is streaming away from the sun at 1,000,000 miles an hour. Again, where does the energy come from? How is it deposited to those particles? And we have a lot of theories, but we really lack observations, really key observations that will help us advance and discriminate between these various theories. So to look at this region, CODEX makes an artificial eclipse. Probably folks saw, earlier this year the eclipse, or maybe in 2017, the solar eclipse where the moon blocks the sun. We do something similar within our instrument. Of course, an eclipse that we see with our eyes may be only a few minutes. We want to do that a lot longer. And look at this faint corona. This is really a key. The corona, this outer atmosphere of the sun, is 1 billion times fainter than the disk of the sun. So this is a really complex process to do that. CODEX in general has 3 high-level goals then: a technology goal, basic science goal, and space weather. And I want to talk a little bit about each of those. First, to make these new observations, these difficult observations I discussed, we've developed on the ground 3 key technologies that we're testing now in space. First is our actual coronagraph design. Coronagraph is the instrument that blocks the sun. There have been previous coronagraphs have flown in space, but we have a new design to really minimize the stray light and really optimize the faint features that we're looking for. Next, we've developed a polarization camera, a specialized camera that can look at the light and separate what's the polarized part, the light that's focused at us in different directions. And this replaces previous missions that have had a very, very complex mechanism. So this is a really lot easier, cheaper, and better way to do it. Finally, the real breakthrough for this mission is the previous missions have used what we call white light, which is broadband light, and they look at the density of material in the corona and the solar wind. That's a key part, but not enough to make our advances in this understanding that we want to do of how this energy is deposited. We need to look at some other properties of the material, of the plasma, in particular the temperature of it, how hot is it, and how does it change with the distance from the sun, and the speed, exactly how fast is it going, and how does that change again with the distance of the sun. So we've developed very specialized filters for this coronagraph that is again very new. So then we use these technologies to do our science. We're studying the corona, which is really the base of the solar wind. Would be the first time that we've measured all three of these key properties: the density, the temperature, and speed of the solar wind at the same time. And we're doing this globally. This whole global picture that you see here, we'll be doing that for at least 6 months or more. So we're really excited about the science and what we're going to learn from it, and how it will feed back into the theories that they're discussed. Finally, there's a space weather aspect. Space weather is what we call the interaction of the sun and our technological society, or even our astronauts' health and safety in space. The sun has these effects, or these high energy particles that can cause interactions. What Codex does is not directly measure the space weather, but these storms from the sun, they're called coronal mass ejections, they travel through this background solar wind that's always there. So just like a hurricane may travel through the air, we need to understand not just the hurricane and the air, but we need to understand the solar wind that the CMEs are traveling through. And that's what Codex is going to do, is provide these new observations of that solar wind. So again, help us eventually in the future to lead to better predictions of space weather. In the future, again, we hope that Codex kind of spurs some thought on the theories, some new observations, even following up from Codex, and really the whole scientific philosophy of we have a theory, we test that theory, we then evolve and that hypothesis and move forward. So we're really thrilled about Codex to do that. You can go to the next slide. Just some pictures to give you an idea. Codex is an externally mounted payload. We're going to be mounted on the outside of the space station. We don't interact at all with the astronauts. Just kind of gives you a size. It's about a kind of a cubic meter or so in size. You see some pictures of during the development of Codex. Kind of the white is the thermal blanketing that you see, kind of covers all of the instrument on that. Next slide. And again, another picture of close near the end, the delivery, some of the final testing that we do on the ground before we launch. And direct everybody. We have a website. It's listed up here. There's a lot more imagery and movies. You can get a lot better idea about the mission as a whole. So it's really Codex is in short. Thanks.
Well, thank you very much, Dr. Newmark. I see that we have a question from our friend Will Robinson Smith.
Yes, hi. Can you hear me all right?
Got you loud and clear.
Thanks, Pat. One quick one for you, Jeffrey, and then slightly longer one. You mentioned the payload will be mounted externally without the aid of astronauts. Will that be done then with the robotic arm on the space station? And then second question, if I might. I know that Codex obviously is its own distinct payload, but will there be some type of collaboration or sharing of data between Codex and the team behind the compact coronagraph that was launched as part of GOES 19? Thanks.
Yeah, both excellent questions. Indeed, it's the Spidim, the robot mounted on the Canada arm that will come over. We are launching on, it will pull us out of the Dragon trunk or the externals. So we'll be in the Dragon trunk. It will pull us out, move us over to the Express Logistics Carrier 3, which is where we'll be mounted on the Zenith site. And indeed, we have in terms of collaboration, we of course have our internal project, but we hope the plan is hopefully the data is going to be really exciting data, and we certainly plan on opening it up to the public. We're going to put it on a community website, and not just the compact coronagraph that you alluded to, which is just launched, and I just saw some awesome pictures from it that were just the other day. But there are a number of other missions that are really exciting. Parker Solar Probe flies directly through the corona that we're measuring at about 10 times the solar radius of, and it measures these properties that I'm mentioning, the density, the temperature, the speed directly. We're focused in on between where Parker can go and the sun's limb, so lower down. And so we very much are interested in collaborating with Parker Solar Probe. Additionally, there's an ESA European Space Agency NASA mission called Solar Orbiter, which has some really exciting measurements that are very collaborative with us. So again, we do absolutely hope to collaborate with the broader community, and our data will be made public, after we validated and everything, hopefully within a month or two of launch.
Fantastic. Dr. Newmark, you actually you answered a question that I was just going to pose to you, but another one did kind of spring to mind, where you did mention the level of collaboration that's kind of taking place, where it's not just your team, but it seems like you're working with a lot of teams across various space agencies and whatnot. Could you maybe describe kind of that process and this, how this level of collaboration has really helped to contribute towards this project?
Sure. There's two aspects that I think I'll highlight very quickly, then let others dive in. One is the development of Codex. Meghan mentioned in the beginning, this was actually an international collaboration just for the development of it. We had key partners in Korea and Italy that were part of this. So that was really exciting, working with those missions, those people there. That was kind of from internal out. We reached out to them and we collaborated with them and help us development. This future collaboration is a little bit more what I would call ad hoc. We haven't. It's not as formal. We as a community, we regularly have large scale meetings and we talk, and so we don't have a sort of a formal, yes, we're going to work with you. It's more of an informal, yeah, these are really exciting data. How do we get together? How do we compare? Hey, let's work on the science problems together. And so we have these more informal contacts that we broaden out.
Awesome. Well, I understand that you are quite a busy bee right now. I know that it was very helpful for you to take a few minutes out of your schedule to join us today, but I also think that you have to run away after this. So if there are any final questions from the members of the media to Dr. Newmark, I would advise to toss them out now. Otherwise, he's going to leave us. Okay, well, again, Dr. Newmark, thank you so much. Greatly appreciate your stopping by today, and best of luck with Codex.
All right, thanks everyone. Looking forward to it. Bye.
Okay, next up, we will be bringing in Dr. Leonardo Barilaro. He is the senior lecturer at the Malta College of Arts, Science and Technology. As lead investigator for the ISS National Lab sponsored AstroBeat project, Dr. Barilaro will discuss his team's plans to use cold welding to apply metal patches to samples that simulate a spacecraft hull damaged from hypervelocity impact. And he might also talk about some other fun elements that are associated with this project. Dr. Barilaro, greetings from across the pond.
Hello, everybody, and thank you, Patrick, for the introduction. I am Leonardo Barilaro, the space pianist, aerospace engineer and pianist, and AstroBeat, for the first time, wants to investigate a phenomenon that usually is seen as something that you don't want, which is cold welding adhesion. And there is an image that can be showed while I describe it. And basically, we developed a technological demonstrator, a test rig, that hosts on board 4 chambers. This is a very compact experiment, as you can show, and there is also an image about the internal structure. In these chambers, the aim is to test the forces needed to get the cold welding adhesion between 2 metal samples. Here, the idea is to repair spacecraft hull following an hypervelocity impact with a space debris, and in this way to elongate, extend the mission time, plus tackling a problem that will become and is already becoming bigger and bigger, above all for missions in low Earth orbit. So usually cold welding adhesion has been seen as something that definitely you don't want. So literature is very little talking about cold welding adhesion in space. We developed these 4 chambers with a calibrated mechanism to deliver forces, and also thanks to an ohmmeter, we want to measure already when we are on board the International Space Station the quality of the cold welding adhesion. The experiment, once back on Earth, will have further analysis regarding the samples. So this is a collaboration done with the institution that I represent, the Malta College of Arts, Science and Technologies in Malta, and I am Italian, as you can see from my hands moving around. Plus, is a big collaboration with Dr. Mark Wiley, which is the co-author of this idea of using cold welding adhesion to repair damages from space debris, and is a lecturer for the Southeast Technological University in Ireland. Plus, we are also collaborating with SkyUp Academy that helped us to develop this render of the experiment, plus a video, and with New York University in Abu Dhabi, thanks to the support of Professor Andrea Macciò, which is the director of the Center for Astrophysics and Space Science. In parallel with this, there is a space art element connected with the mission. In fact, my music will be again symbolically on board the International Space Station, and there will be a streaming event, actually 2 streaming events, of this music from the ISS on Earth. And this, as you can imagine, is a powerful dissemination activity that helps to bring together space enthusiasts, and also to bring closer to space topics people from different backgrounds. For this mission in particular, I have the honor of collaborating with a world-renowned musician, cellist Tina Guo, and producer Steve Mazzaro, that collaborated with me for in realizing one of the compositions that are together related with experiment. And there is also a photo of me with Tina Guo. And Tina Guo and Steve Mazzaro are also known because since several years they are also collaborating with Hans Zimmer. So it's the science fiction world that is helping the actual science to resonate more and more with, above all, the general public and also space enthusiasts. Our target with this mission is, first of all, to close the part about the space art, is to be able to reach out to a much bigger public. And also with New York University in Abu Dhabi, we worked with students and 2 professors, in particular Professor Carlos Guedes and Professor Matteo Marciano. So we already expanded the activity with students from a different course of studies. Plus, with the experiment itself, we want to have, of course, a successful technological demonstrator. We want to retrieve data that doesn't exist in literature, We want to see also the issues because there will be also metallurgical analysis performed in the post-flight phase. And this will be the baseline to have a sort of Astrobit 2 where we want we aim to develop a sort of proper tool that can be used to actually repair space debris damages. So that's all I have. And if there are questions, I'm happy to reply.
Awesome. Well, fantastic. Thank you very much for that overview. We did get a question from our, this was prior to where we asked the public if they wanted to ask a question, they could do so through the hashtag ISS National Lab. And pretty basic question, but how will these findings impact future missions?
So nowadays, the risk of space debris is growing more and more. Is quite famous now for who works in space, the risk of Kessler syndrome. So missions in low Earth orbit are becoming more and more, and also are in the work new private space stations and also more private missions. So with the increase of the risk on one side, the increase of missions, having the possibility of doing in situ repairs is something that is still not available. There are some few historical examples, like with I forgot his name, a Russian astronaut in on a Soyuz mission that to repair a damage from space debris used basically a sock with epoxy resin. So that was a really do-it-yourself makeshift solution, but also there aren't still procedures. So space debris, there are a lot of topics, so actival space debris removal, better shields, that is also my field of research. I'm working on the 3D printed shields, carbon fiber. But another way to mitigate the risk of space debris is with repairing techniques, and this can also help for deep space missions going, for example, to the moon, Mars, and beyond the asteroid belt.
Fantastic. And so maybe is it possible also to describe kind of the facility itself? I believe that this is kind of an autonomous platform, where one that doesn't have a lot of astronaut interaction with it. Could maybe talk about the configuration of how this project is going to be housed?
So, so excellent point. Astrobit is a fully autonomous, actuated remotely experiment. So we have our unit that communicates with the communication interface, and it will be operated in Houston by the team of Voyager Space. And basically, our target is to have the different chambers actuating in different days. And basically, there is, to put it in simple terms, a stepper motor that will actuate the different spring-loading mechanisms. And we can act on also the number of revolutions and parameters. So we want to have one test, download the data to have a preliminary analysis. So we have also a pico camera. So we want to see that the actuation was performed properly. Plus, there is the idea of an ohmmeter to read the resistance on board. So we will have preliminary data to see how the actuation went. And based on that, on the second day, we will do the other actuation and so on. So there is the fully automated part. We hope that the experiment will be already successful in terms of preliminary analysis. And as I was mentioning, there will be all the part of the post-flight data analysis, where we would use a much more precise ohmmeter, and also there will be metallurgical analysis. So here the idea is to decouple the effects. We want to see, to measure, because it's mainly a metrology and measurement experiment. We want to have a confirmation of the forces that we are expecting to have a proper cold welding adhesion. So basically we have a male and female pin that will be put in contact with the interference. And basically between the two components, we are aiming at having the cold welding, and in this case, sealing the hole. Of course, this will not be the setup for a real space debris hole. This is to start measuring the forces and having an idea of how bad or how good behaves the samples that we calibrated before sending them to the ISS.
That was a great, great overview, great and fantastic response. Well, again, best of luck with this investigation, and also on top of that too, best of luck in becoming furthermore the space pianist. So that is quite the
Yes,
juxtaposition.
my target is to follow example of Sarah Gillis that a few weeks ago did an amazing feat with the violin. My detail is that I want to do it with a piano, and step by step I'm working to get there and stream a concert from space.
Bringing different space missions together. I love it. Well, thank you again. Greatly
Thank you very
appreciate you
much.
joining us today. And we're going to move on now to Dr. John Barnett, who is the President and Chief Science Officer for ExesaLibero. Although I'm sure I botched that, and I am looking forward to finding out how you're properly supposed to say that. But Dr. Barnett will highlight how and why his company aims to leverage microgravity to crystallize proteins associated with its novel drug known as ELP-004, which prevents excess bone removal associated with numerous diseases, most notably rheumatoid arthritis. Also joining Dr. Barnett is Rich Bowling, who is the VP of Corporate Advancement for Redwire Space, the hardware partner for the team on this project. Gentlemen?
Thank you, Patrick. Really pleased to be here and express how we're utilizing the space station on this exciting project for ourselves. And so to get started, let me give you a little primer on bone biology, because this seems to be a point that a lot of people don't understand. So when you become adult, a lot of people think that your bone is fixed and nothing else happens to it, of course, unless it gets broken or something to that nature. But in fact, that's not true. What happens in normal life is that bone is constantly being broken down or dissolved, and new bone is constantly being built in its place. So ultimately, then there's no net effect, and you don't feel any differences in this occurring. And this occurs through two different kinds of cells that occur in the bone. But when you have some kinds of diseases like rheumatoid arthritis, osteoarthritis, and some of the cancers, what happens is that the cell that causes the breakdown of the bone, which is normally in balance with that's built, actually gets too active, and it breaks down more bone than can be built by the other cell type. And so as a result of that, what you actually get is the pathology associated with rheumatoid arthritis or osteoarthritis, which, as you know, can be painful joints and that sort. Can we have the first slide, please, Patrick? Thank you. So I'm John Barnett. I'm the president of this company, a small startup company called ExesaLibero. And we're in Morgantown, West Virginia, and we're developing a drug, which we call ELP-004, that prevents the bone lesions commonly associated with several human diseases. And that includes the rheumatoid arthritis and other types of arthritis, but it also includes several types of cancer. One of them is multiple myeloma, which is actually a cancer of the bone, breast cancer and prostate cancer, which are very, very common, also metastasize to the bone. And when they do so, they cause the bone to get brittle and break down. And so that's a very painful procedure. Also, trauma-induced arthritis is a problem. Trauma-induced arthritis is the leading cause for our military people to be off to actually I'm losing that train of thought here, so that they are no longer able to be in the military because of this trauma-induced arthritis, either because of contact injuries or because that they have done something in the normal line of duty. But trauma-induced arthritis is also a problem for people that are doing sports types of activities that involve a lot of trauma with that. And finally, space travel, because in space travel, almost immediately when space travel begins, you get loss of bone thinning. And so there's not a really good way to treat that going on. May I have the next slide, please? So our drug is really has great properties. What you see here on the two columns on the right-hand side of this slide are the two types of treatments that are available to treat these types of things. One is called the bisphosphonates, and most of the bisphosphonates are administered intravenously, but there are some oral ones, but these have terrible side effects. They cause fever, flu-like symptoms, hypocalcemia or a loss of calcium, and bone and joint pain. But one of the major ones that is really a problem is something called osteonecrosis of the jaw, or ONJ. This is a really horrible disease in which when people take bisphosphonates, and usually as a result of having some sort of dental pathology at the same time, the whole lower jaw gets degraded. And of course, that results in a loss of teeth, etc. Bisphosphonates are expensive, and so that means it's a high cost to patients. And also there are biologics, as we call them, and these target the immune system. Examples of that are things like Humira and etanercept, which actually inhibit parts of the immune system, causing an immunosuppression of the actual reagents that cause bones to be degraded. And these are administered by injection, which means that you have to have cold chain custody, which means somebody has to be available to receive the package each month. And they're very expensive, high cost to patients. In contrast, our drug, which is showing on the far left column, has very few side effects. It acts directly on the cells that cause the bone to be degraded, and it's not a plus or minus. We can actually titrate it. So actually you can reduce the amount of activity by the osteoclast, and by doing that, we bring it into balance with the cells that build the new bone, which are called osteoblasts. So this means that it can be used for prophylaxis, and it's not immunosuppressive. So you can imagine that astronauts, for example, that have problems with bone erosion, and that they could take oral administration, a pill each morning, for example, and this would prevent this bone erosion from occurring, or this bone thinning. Our drug is produced by synthetic chemistry, and therefore it costs pennies to produce, which should result in a lower cost to the patient, and therefore for those people that are interested in developing this, a high profit margin for that company. May I have the next slide, please? So what is the need for these types of things? Well, rheumatoid arthritis, there's 53 million total patients in the United States with rheumatoid arthritis, and that's projected to actually go up to around 70 million patients in the next 10 years. Multiple myeloma, which is that bone cancer, there's about 35,000 new cases per year. That doesn't even include the ones that are still here. And there are other indications which, unfortunately, have come out. I didn't notice this, but didn't get translated very well in the slide, getting translated to a PDF file. But the first one that should read osteoarthritis, and about 1 in 12 U.S. Adults over the age of 70 have symptomatic osteoarthritis. So they're projected to be about 325 million total osteoarthritis cases in the United States. Osteoporosis, which is really common in women, which about 27% of women have osteoporosis, and about 6% of men, of those over 65, have osteoporosis. And then there are several bone-seeking cancers, which I mentioned earlier, one being prostate, which is very common in men. About 1 in every 100,000 men have prostate cancer. And then breast cancer is very common in women. About 119 women per 100,000 have breast cancer occurring. I've already mentioned that degenerative or trauma-induced arthritis, which is the leading cause of unfitting conditions for U.S. service personnel. But if you're looking for other uses for this for the civilian population, athletes who do athletic tasks that involve a lot of activity have problems with degenerative arthritis as well. And finally, I wanted to talk about the bone thinning caused by microgravity. And I think, in fact, it's not unreasonable to assume that at some point in the near future, there's going to be some space travel to go on here. Either some people have ambitions to actually go to Mars, which of course is going to take months and months. And so you have to have something that's going to prevent this bone thinning that occurs. And the bisphosphonates and the biologics don't have the right capability to do that. And so that very anxious to their develop this drug for that purpose at least. May I have the next slide, please? So I wanted to show you how it works. You can see here two groups of pictures. If you start with the one on the right-hand side, the group of pictures there, which I show a vehicle, that means that the drug, whatever the drug is dissolved in, is given to an animal. And this is a mouse, which has developed arthritis, and you can see where the arrows show there's tremendous amount of bone erosion that occurs in this mouse's paw. And at the lower part of that slide there, you can see a close-up of that area there, which shows areas in which have just been dissolved away. If you look on the left-hand side, that group of panels there, we've done there is we've taken an animal and we've given it human multiple myeloma cells. So they develop bone cancer. And you can see that the bone on the left-hand side has no lesions in it or no degradation associated with it, whereas the one on the right-hand side has big holes within the bone, which is actually extremely painful to patients and needs to be developed for that reason, if nothing else. So we're really excited to be able to be included in this project because most of the knowledge on chemical synthesis is done at normal gravity on Earth, or 1G, and that, of course, is very constant. Whereas when we actually are able to do this in the International Space Station, we'll be able to do this in microgravity, which we can see whether it makes a difference in how the drug is produced, and see whether or not it's actually more effective when it's produced in space versus when it's produced on land or on Earth. So lots of things, really excited about that. Excited to join with Redwire, and I think Rich is going to tell you about the device in which is used to actually do the experiments.
Let's see, do we have a picture of that, Tim? I think we may have one here. There we go. Okay, so what you're seeing, the light blue boxes, those are what we call pill boxes. And of course, with space, everything is an acronym. I'm sorry, I didn't come up with this one. But Pharmaceutical in Space Laboratory, PIL is the name of our system and our program. The box stands for Biocrystal Optimization Experiment. So there you get pill box, and that's what those, physically we call those blue boxes. And that is what the ExesaLibero payload and processing will take place in. We have 11 payloads of different sorts on board the International Space Station, but the most of what we've been processing in bulk has been drug manufacturing, and we really want to thank Dr. Barnett for his partnership on this mission. Thank the ISS National Lab for helping us with the upmass and the crew time. Crew time is very limited with this sort of investigation. Essentially, we just asked the crew to remove these cassettes from the cargo ship when it comes up and insert these cassettes inside of the microwave-sized lockers that you see behind you there. There's a couple of our Redwire lockers there behind the cassettes. It's essentially automated. We control it basically from our own little mission control on the ground. So we also want to thank NASA for its support in helping us develop this system through its in-space production applications program. We've been working on developing the pill system and these pill boxes for about 4 years. And we have just gotten into the operational phase at the end of last year. So in total, we've flown 16 pill boxes in space for a variety of customers. And on this mission alone, we'll add 12 to that number. So again, we're grateful for all the partners that have helped us develop this capability, which we are seeing great demand for from industry. We're excited to use the ISS as a terrific incubator, not only for technologies such as Pillbox, but really also for the business models that we expect to carry over into vehicles following the International Space Station, vehicles provided by commercial entities. And we're excited to be a part of those plans with those partners on into the future. So pillboxes will be a part of certainly our strategy for low Earth orbit commercialization. So thank you very much for the opportunity to talk about Pillbox and Pill and Redwire. And again, Dr. Barnett, really grateful for your partnership on this mission.
Awesome. Well, thank you both very much. Quick question on my end. So if Dr. Barnett, if this project is successful, could this possibly speed up the ability for, a treatment like this to reach FDA approval, as an example?
Oh, yeah, we're very excited about that part of the possibility as well, because as you can well imagine, for a new drug, we have to go through clinical trials for this approval from the FDA. And these, of course, diseases like rheumatoid arthritis and osteoarthritis are very prolonged, and it'll take a good deal of time in order to prove that our drug is effective. Well, if we can actually treat astronauts with the drug as they go up into the space station, the bone thinning that occurs in these individuals occurs almost instantly when they get into space. And so we'll be able to determine very quickly whether or not the drug is effective or not. And that should speed up the actual approval of the drug.
Fantastic. I see that we have a question from our media. Allie?
You hear me?
Gotcha.
Hi, Patrick. Really grateful to both Rich and John for their outstanding R&D work. The question is that for this mission on the ISS, are we testing the ELP-004 for two possible applications for melanoma and rheumatoid arthritis, or are there any other aims of this study that will be targeted for this mission?
Great question, Allie. In matter of fact, in this mission, what we're actually testing is the actual synthesis of the drug to see whether or not producing the drug in microgravity makes a difference, and whether the drug is more effective than if we produced it on 1G gravity or on Earth itself. And so that's the purpose of this particular mission. We're hoping that we've applied for a grant from NASA to actually test the ability of the drug to prevent the bone erosion that occurs. And that will be done in petri dishes, or in vitro, as we call it, rather than in animals. So that's we hope will be the next mission.
Good luck.
Awesome. Well, thank you very much. And again, equally looking forward to seeing what happens with this investigation. And Rich and to the Redwire team, continued success with Pillbox and bringing in some great partners along the way. Next up, we would like to present Dr. Prashant Nagpal, who is the co-founder and chief operating officer for Sachi Bio. Dr. Nagpal will provide an overview of the ISS National Lab sponsored Rapid Low-Cost Drug Delivery in Space project. This investigation will test cutting-edge RNA therapeutic, and I'm going to probably botch this one, but nanoligamer technology using brain organoids created using cells from healthy adults and patients with Alzheimer's, Parkinson's, and dementia.
Thank you, Patrick, for the kind introduction. If you can switch over to the slide mode. Thank you all for joining this morning, afternoon, or evening, whatever place you're joining from. I'd like to start with some acknowledgements. So thank you, ISS National Lab and NASA's In-Space Manufacturing INSPA program, and obviously the taxpayers of the United States for allowing us to embark on this really amazing project. So a lot of pharmaceutical companies, virtually every single biopharma on this planet, aims to bring some cutting edge new technology directly to patients, because there's a lot of patients that suffer from these debilitating diseases, some of which Patrick mentioned, and the ones that we will address on this specific CRS-31 mission relate to amyotrophic lateral sclerosis, ALS, Alzheimer's, dementia, and cancer, but in specific glioblastoma multiforme, or a very, very aggressive form of brain cancer. So a lot of companies have been looking at new technologies or development of new technologies, but as you might be aware, neuroscience and in particular neurodegenerative diseases have been a major, major hurdle. At least in the US, the last Alzheimer's drug that was passed by the FDA after a lot of opposition came after a span of 23 years. And even that has now proven to be, not as effective and is being pulled out from the market. So one of the reasons that companies have looked to space as one of the key frontiers is because a lot of drug discovery has been done on animals, which are not good surrogates when you're going to spend billions of dollars into, not only identifying a drug, but also translating it. And space provides one unique opportunity where a lot of these neurodegenerative diseases and carcinogenesis, cancer, gets accelerated and the clock turns it really, really fast. So if you'd like to know a little bit more about our program, the first link down at the bottom provides our information about how we've really advanced this space-based drug discovery. Sachi Bio has used a very innovative in-house technology to really utilize this opportunity to accelerate drug discovery for a number of neurodegenerative diseases. And the second link provides an American Chemical Science, ACS, Chemical Neuroscience publication on sort of the scientific details. So if you could move on to the next slide. What we've done here is our technology platform is a very, very cutting edge RNA therapeutic modality. So the big problem, as I mentioned, it's not like people don't want to bring new innovative technologies. One of the key hurdles is more than 85% of proteins within the human body. Many of them are high value targets for cancer and for neurodegenerative diseases, some of which I list here, are still considered, after spending more than $280 billion each year on pharmaceutical R&D, are still considered undruggable. So what Sachi has done, obviously, obviously utilizing this massive opportunity that has been provided to us once on SpaceX 29, is really opened up the remaining 85%. So everybody was playing in this 15% sandbox, and we've come in with our nano oligomer platform, use not just this very advanced RNA therapeutic, a peptide-based RNA therapeutic, but also our delivery system to now open the field, and virtually every single protein, every single target, however hard it was in the past, has now been opened up for not just discovery, but also for translation. And the way we do this is we are not going after simply proteins or small molecules. That's where the entire biopharma is highly focused. But we are sort of cheating a little bit, going one step over and trying to see how human body actually produces this protein. In our overview, it was very difficult to make these human proteins outside the human body. So we are just using cues, essentially, or mRNA and DNA, to be able to produce this. And our nano oligomer molecule does that. What this really allows us to do is really go after disease causality. Rather than just going after symptoms, we are going after the causes, and doing this in a massively high throughput way. We don't test one drug, two drugs. We're testing more than 100 drugs to bring you that one best-in-class and first-in-class molecule that shows remarkable efficacy in one, two, or more diseases. So what we've really shown is you can drug potentially the undruggable. And on sort of the mechanics side of it, how we've done this is we've ensured that we can selectively, this drug is highly potent, highly targeted, but we can, you can have the best drug in the world, but if you can't deliver it to the right organ, it's of no use. What we've shown is you can really deliver it to the tissue, to the organ that it really is needed in. So we've shown delivery to the brain, different sections within the brain with very, very high accuracy, and with multiple routes of administration. The same drug can be an oral pill, can be an injectable, can be an inhalable route, and we've shown all of them, obviously, with different efficacy into each organ. And we've really shown the world's first oral RNA therapeutic to be very, very effective for inflammatory bowel disease, and this drug is going to fly. It's shown extremely high level of safety. We've done sort of missenses where we've sort of scrambled the sequences. They don't match anything within the human body, and it just simply passes through the urine. Next slide, please. So what we've really done is combine this amazingly precision and high delivery ability of our nano oligomers with a lot of these patient-derived organoid models. As I mentioned, one of the key hurdles was a lot of this early drug discovery was being done in animals, and we can try to mimic the human physiology within mice and rats and dogs, but it's not quite the same. So what we've done is we've taken actual healthy and diseased patient organoids, in this particular case, brain organoids. The entire payload is full of, brain organoids from different patient populations that represent that particular disease. So there's, for example, for ALS, there's sporadic ALS, C9 open reading frame ALS, SOD, and TTP43 ALS. So virtually all causes are represented in there, along with healthy 3 or 4 healthy individuals. And then we're using the microgravity acceleration that has been shown now with our prior flights that massively accelerates this pathology. And what we are seeing is we can screen a lot of different best-in-class and first-in-class lead molecules to massively downregulate just within one flight from hundreds of these top targets and very potent molecules to that one molecule that shows the highest efficacy. This translates into both a really good opportunity for the patients and massive translation through this preclinical and hopefully the clinical process. Our clinical trials are slated to start next year, but also as a great business opportunity to cut down the early discovery time, which accounts for more than 60 to 70% of the entire timeline, and bring really cutting-edge technology and therapeutics to the patients. Next slide, please. So talking very specifically about the SpaceX 31 payload, the CRS-31 mission payload, in here we are flying more than 1,200 patient-derived and healthy brain organoids. 588 of them, due to space constraints, of course, will be flying to the space, and more than 640 of these concurrently made with the exact same patient at the exact same time will serve as synchronous controls. And in this particular flight payload, we are testing or conducting virtually a parallel clinical trial for amyotrophic lateral sclerosis. As many of you might be aware, it's a very horrible and a debilitating neurodegenerative disease where there are no cures, no therapies. Alzheimer's disease, Parkinson's, and glioblastoma, specifically in using this massive carcinogenesis that's been observed in microgravity in our prior space flights. We've been able to use these models in both healthy and diseased patients. So we have comparative testing abilities for all of them. And from the nanoligamer, the high throughput discovery platform side, as you could see, we are testing 9 targeted drugs in this particular payload on these 4 different diseases. Some of the drugs, as you might see from the infographic here, are identical. For example, our blockbuster drug NI-112, or SB-NI-112 as we call it, has shown remarkable efficacy across a range of different neurodegenerative diseases, including ALS, Alzheimer's, dementia, Parkinson's, and also other autoimmune diseases like inflammatory bowel disease, rheumatoid arthritis, and others. It's an extremely potent drug that targets the body's or the dysregulation of the immune function within the human body to target a lot of different diseases. More than 80 conditions, as you're aware, are caused by just a dysregulated immune system. And a lot of them we've tested with this one single drug, and we expect it to be a big blockbuster. So thank you all for your attention. I wanted to keep this very brief, and I would love to answer any questions that you guys may have about our payload, about the platform, or what we are trying to accomplish in this great opportunity, thanks to ISSNL and NASA Inspector.
That was fantastic. So 2 quick questions that popped into mind for me. One of them is maybe more a little bit about the project itself and the facility. I believe that it's going to be leveraging a Space Tango Lab, correct? And maybe kind of highlight what's unique about that. The fact that there's really not a lot of astronaut interaction with this, but again, that there's a lot of interesting engineering, I think, that is trying to assist your investigation.
So because we intend for these investigations to be directly translational, so we are in discussions with FDA. What we do at Sachi is each of these are individually packed. That's why there's not much interaction with the patients. And we what we are trying to do is establish a GMP process and an approval process with the FDA, where the drug, obviously the surviving medium, whatever food the cells and the organoids need are pre-packed into a sealed vial, which are put in an incubator and taken aboard the space station. They're brought in back to Sachi. So there's a chain of custody that's maintained throughout the entire process. Again, keeping in mind, because a lot of the investigations, especially early phase R&D work, sometimes uses a lot of interactions. That process does not go really well with the good manufacturing practices or good laboratory practices that are directly translatable. So since this is a very expensive endeavor, to actually take all of these organoids to space, and these are very valuable samples. So Sachi has developed this process for potentially direct FDA approval. Now, one of our drugs, as I mentioned, is going to go into the clinical trials next year. So we are using that alongside an established paradigm of just animal-based testing. But then we are putting our other, as I mentioned, 9 innovative drugs alongside. And our hope is that that early drug discovery process will be more humane, number one, and not rely on needless animal testing, where animals, are used for testing of this drug and then sacrificed, and actual human sort of organoids can be used for this process, and also to have higher clinical translatability, because that is the other key endeavor. Just it is research, it is early phase R&D, and as we said, the future is already here. The space-based drug discovery from some of our investigations is already becoming a reality, as you could see in some of the other investigations that ISS is also sponsoring. But we want it to be a one-to-one correspondence in being able to take this very precious samples and get clinical approval based on that.
Fantastic. And I think that from my perspective, it's really fascinating to think that space in some ways could also maybe potentially help to accelerate kind of these drug designs for patients. And so I guess the last question I would ask is, what kind of got your team excited about the notion of wanting to do something on the space station? Kind of how did that process arise?
Absolutely, and that's a great question. So, as I mentioned, a lot of these diseases, drugs come out after spending billions and billions of dollars, and ultimately in clinical trials or in actual testing, it fails because they're number one tested on animals, which are sometimes surrogates, but sometimes maybe not perfect models. The second part of it is the heterogeneity, and that's another important feature that the space brings. Sometimes the early testing is done, even clinical testing is done on a subset of patients, not quite representing the entire disease pathology or the entire population that suffers from this disease. So a lot of time people may be popping pills and taking these very expensive drugs when it actually may not be providing any physiological benefit to them. So Sachi was always formed with a goal that we need to have therapeutics that are physiologically active and actually provide relief from these debilitating diseases, rather than just relying on metrics that people use in terms of money and sellability. So from that perspective, doing this early phase drug discovery in a number of different patients, so we really bring out the molecules, because we have more than 100 molecules that show remarkable efficacy. How do we bring out that one molecule that would give a large set of representative target population, age diversity, gender diversity, racial diversity, ethnic diversity, so we can have maximum relief for patients? So that's always been the mission of our company, and that's really been enabled by this, because now we can really accelerate the pathology for all of them, rather than relying on one or two model systems or model cell lines that are readily available. So that's where the space-based drug discovery has been very transformative, and hopefully would be soon we'll be able to make it available to patients.
Fantastic. And one thing that you alluded to, and also that Dr. Barnett did too prior, is that again you're looking at actual drug targets themselves. And I think that that's what's really exciting too, to see the evolution of how things have gone on station, where it used to be very fundamental in nature, but now, teams like yours are really trying to apply, drug targets that you're actually actively developing, so that way they could be better for patients on Earth and beyond. So that's fantastic. And I wish you and your team the best of luck, and looking forward to hearing about the success of this investigation down the road.
Sounds good. Thank you, Patrick. Thank you, everyone else.
All right. Well, last but certainly not least, we have Dr. Agata Zupanska, research scientist at the SETI Institute. Dr. Zupanska will highlight the NASA-sponsored Artemis project, which will examine whether Antarctic moss recovers from the impacts of cosmic radiation and microgravity, and if so, how? Dr. Zupanska, good afternoon. Please tell us a little bit more about your investigation.
Good afternoon. Thank you, Patrick, for the introduction. So we can turn now to a very basic fundamental plant biology, because this is what Artemis is about. May I please have the first slide? Thank you. So I represent a team of Artemis. So this is NASA, SETI, and University of Florida collaboration. And Artemis aims at understanding of what are the effects on plants of the two space environments in combination. And that is the cosmic ionizing radiation and the spaceflight microgravity environment. Can I please have the next slide? So we know very big amount about how plants behave in microgravity. We do not know nearly as much about how plants do in cosmic ionizing radiation as it is in deep space. What we absolutely do not know is how plants respond when they are challenged by both of those environments, which of course is going to be the scenario when plants are on the interplanetary travel or they are forced to grow in lunar or Martian farms. So may I please have a next slide? So for this investigation, we are using moss, and there are a few reasons for that. So not only moss is a very good gravity sensing system, but moss is the most tolerant of radiation among plants. It can not only survive, but really thrive under doses and energies of radiation that are lethal to other plants. And we brought this a little bit further because we selected Antarctic moss. So moss that grows in Antarctica is challenged pretty regularly with high levels of UVB, or at least during the 3 months of ozone hole. So UVB, UVC normally do not reach the Earth's surface, but in Antarctica, because of the compromised atmosphere and ozone hole, there is high levels of UVB that this moss has to grow. But so it has this tolerance to the naturally occurring UVB radiation there. But it has to also respond to a lot of other extreme environmental conditions. So Antarctica is really the harshest of the terrestrial environments. So Antarctic moss needs to deal with the extreme cold, extreme aridity, extreme, high albedo, and of course, wide temperature ranges, which I think is even worse. So basically, by selecting Antarctic moss for Artemis investigation, we know that we have in hands not only plant that is very highly tolerant of radiation and should really manage and do very well under condition of deep space ionizing radiation that is pretty damaging to biological systems. But it's also kind of used to be challenged by other extreme environments. So we think it should do also well when we force it to grow in the spaceflight microgravity. So can I please have next slide? So how we execute our Artemis. So we start with the ground portion of the study, and this is quite important because we are irradiating moss with ion beams, and by doing that, we are simulating this deep space cosmic ionizing radiation environment. So we cannot use radiation that is on the International Space Station because although there is higher radiation than on the surface of the Earth, that radiation is not as severe as it is in deep space because there is still protective effects of magnetosphere on the station. So we use accelerator at the NASA Space Radiation Laboratory at the Brookhaven National Laboratory, and we basically bombard our moss with ions that are accelerated to the relativistic speed. So we really recreate this condition in which moss is under condition of the deep space ionizing radiation. And then we do not allow moss to repair any radiation-induced damage by putting moss into cold temperature. So we keep our moss at the 0.4 Celsius and not above 4 Celsius, which turned out to be really the biggest logistical challenge for Artemis execution. We take it to Kennedy. All that time is moss in cold storage. And really, moss only when it's taken by the astronauts from Dragon and placed on the station in ambient. This is when moss warms up and can start its normal physiological processes, and it has finally a chance to repair whatever damage was caused by that radiation simulated on the ground. So this is our strategy to really force those two space environments on the plant and see, can it really repair this ionizing radiation induced damage when it's in the spaceflight microgravity? Is it really efficient? Those mechanisms that it hopefully already developed while growing in Antarctica, can this utilize in space, and how well it's really doing? So once we understand what is that this moss is doing regarding mitigation of the radiation hazard in the reduced gravitational environment, we can then look at those fundamental processes and enhance them in other plants that are important to our missions, mostly those that give us some food. So space crops are most likely candidates. But we can also know that plants from the extreme terrestrial environments can have some edge in doing pretty well in complex, space environments. Can I please have the next slide? So basically, I would like just to highlight the most important things about Artemis, just in a nutshell. So for the first time, we are getting Antarctic moss to space. That in itself is quite exciting. For the first time, we are really having this opportunity to look how plants are gonna cope being challenged by two space environments, pretty severe ones, like cosmic ionizing radiation, that's a hazardous one, and also this very novel, space-like microgravity environment. And I think the major strength of Artemis is that we are doing all of that in living plants. So yes, we are inhibiting moss metabolism by keeping this in cold, but this is a live plant that is experiencing irradiation and then has a chance to repair whatever damage is caused in the reduced gravitational environment. So the previous studies were, for instance, looking at the seeds that were irradiated and then germinated in some simulated microgravity, like on a clinostat. So although valuable, obviously, it answers slightly different question than just looking at this physiology of the plant that is growing in those, very unfamiliar space environments. With that, I'll be happy to take some questions and talk some more and expand on why Artemis is really so cool.
Artemis indeed is quite cool. And if I was a betting person, I would assume that maybe the name of this derived from another mission that NASA's pretty high on.
That's exactly Artemis. We were like, how are we gonna do it? We came up with the Artemis first. It just sounds so good. And then we work out like how it can be an acronym and that stands for what. And that took us a while. And it's
almost like we live in a world of acronym soup here over at in the space community, right?
Yep. But Artemis contains, there is the moss, there is already indication that there is something about moss, and lovely. And of course, Artemis. Besides, we are preparing our Antarctic moss for lunar missions, hopefully one day. So yeah, there is the connection there as well.
There we go. So one of the questions that kind of popped up while you were chatting about this, and you alluded to it a little bit, but are there some other examples of how this research could contribute to bioregenerative life support or something of that nature?
Yes, so I get back to this very fundamental, biology. Again, as a space biologist, we just don't know what happens to those plants. We do not have the data on seeing what happens when the plant is challenged in those two environments. So what I believe we will learn from Artemis is that not only the plants, some plants, at least, moss, are capable of repairing whatever the damage is caused by the radiation. They can do it in the reduced gravitational environment. I don't know if it's gonna be as effective. I would expect that there's gonna be some compromise there, some processes, but we hope to really find the mechanism and the processes that do work and make a plant tolerant of those two environments. So then we can, because we are going to be looking at the genes and proteins, a lot of molecular data that we can come through, and we can identify which are those pathways. And then there is this process that we can or enhance this in other plants, so they can be better fitted for, being grown as crops and food plants. Or we can just genetically engineer, just transfer genes and make the plant, like what is behind this tolerance of that radiation and what is that enhances growth in the microgravity. So that is the first thing. So just the direct translation to other plant species. But there is also one other aspect. If we really see that moss is doing very well in those two environments, there's of course many other aspects of being in space, be it in travel or being in the lunar environment. And there is still a lot of other parameters that plants will need to survive also. But we think that moss is a particularly well positioned for space exploration. It's a very small plant. It's a low maintenance plant. It grows on everything and any sort of substrate. You will have moss. Yes, just please go outside your building and see where moss can grow. Propagation of moss is absolutely ridiculous because you just break off the filament and place it on whatever substrate and it just keeps growing. And it's really hard to kill. I can attest for that because I can neglect my moss for months and months, and it's still there, and it's just a little brown and yellowish, but it's like, yeah, I'm hanging in there. So it's a remarkable organism. It's also a pioneering species. So it has those ability to really transform very poor, very strange ecosystems that other plants just cannot grow in. This is the first plant to colonize ecosystems that were ravaged by, wildfires. It's the first plant that colonized Chernobyl after, the nuclear plant accident. So this plant is really just the first one to come in and start transforming ecosystems so they are more conducive for other plants to grow. So we actually see this use of Artemis for instance, enriching lunar regolith. It would be quite beneficial to take some moss to the lunar farms and just make it help not only using soil building capacity of moss, but then of course you can start composting moss and giving some organic matter, so we don't need to take kilograms of fertilizers and nutrients. So there's a lot of other things, and then moss is also very skilled in photosynthesis, and this is just the 417 million years of evolution, yes, when you have to photosynthesize in very different environments and different conditions. So high CO2, other plants just cannot photosynthesize. Moss is just perfect. It actually regularly has pretty high CO2 levels when it sits on the trunk of the trees. Yes, it's like that. There's not that much. So moss has really, besides being this very, in a sense, simple yet very fundamental system for looking at the biological processes that are occurring in plants. So we can learn about those processes and engineer like plants, other plants that are more tolerant of space. We can also use moss, is particularly if it turns out that it outperforms, many other plants in space, and it just doesn't care. That it's been bombarded, with ion beams that are just killing other plants, yes.
Well, and I think to that point too, there's been a lot of different types of moss that have gone to space, I want to say, in the past, and I want to even say fire moss has gone to space. So really, you have Antarctic moss on this one, but then you also have things like fire moss. So it just demonstrates, I think, to your point, that this is something that we very prevalent and something we want to learn more about.
Yes, actually, fire moss was the first moss that was taken to space. It was during the space shuttle program. It went on 2 flights, on Columbia flights, and actually the last Columbia flight, STS-107, had cargo with that very moss. And it's remarkable, but they found moss in cassettes in the payload scattered in over Texas. They retrieved some of those samples, which is amazing. So those studies back then weren't really focused that much on molecular aspects, just because there wasn't this technology, this robust, cheap technology that we can do any kind of omics we want from, one moss filament, yes, like just. But they were looking at the very fundamental again, processes is like how can Mars even sense that is not in the gravitational, it doesn't have the, like the physical reference to the gravity vector or what happens. And everybody was betting that Mars is gonna just be tangled up, it's gonna be completely random, it's gonna be confused, we'll have no idea which way to go. And then Mars came back from the station, forming beautifully, clockwise spirals, every filament nicely aligned, just forming the spirals. And it's like, wow, plants do feel. This is amazing. And they rearrange their growth patterns to whatever, environmental cues you give them. Yes, so you remove gravity, okay, fine, I'm just gonna do spirals right now. If you, of course, we can modify a little bit of the plant architecture or growth in microgravity by using light. If we do the strong directional light, we can correct a little bit and make it more directional growth, because then light becomes this dominant, environmental cue for plants. But yeah.
Well, I will tell you what, we need to bring you back on because it sounds like we can talk about moss all day long.
Yeah. I've been very sad that I only have a few minutes for this presentation. I was just like, I can take an hour, please.
Well, we can always do that next time. So in the meantime, best of luck with this investigation. And
Thank you.
Again, we'll chat more moss in the future.
Thank you so much. Glad to be here.
Well, this has been a lot of fun on my end, and hopefully it's been informative from your perspective. But I also, again, I want to thank all of the panelists for taking time out of their busy days, especially those who even joined us from across the pond. I also want to thank the media who took the time to join us for today's webinar focused on R&D launching on board SpaceX CRS-31 to the space station. At this juncture, I do want to toss it back over and see if there are any final questions from members of the media. If not, then we'll get ready to wrap. Okay. Well then, as mentioned, we are looking at a launch in the coming weeks from Launch Complex 39A from NASA's Kennedy Space Center in Florida. For up-to-the-minute information, please follow at NASA, at space _ station, at NASA Kennedy via social platforms, or follow the coverage on NASA's International Space Station blog. And again, note that the ISS National Lab team will have a variety of content going live over the coming days on projects that we are sponsoring. So with that, hope again that you found today to be very informative. It's been a pleasure to moderate today's webinar. On behalf of the ISS National Lab, I'm Patrick O'Neill. Go Dragon and go Science. Goodbye.